Good afternoon, everyone. Welcome to the WALS Wednesday afternoon lecture series today. I am Grace Huang. I am a program director at the National Institute of Neurological Disorders and Stroke, and I also work for the Brain Initiative. I'm delighted to be here today, but before we get started, some very important information for those of you who are here for continuing medical education credits. The code is, look at that, 57659. And if you are attending virtually and have questions, please hit the Send Live Feedback on the videocast page, and your questions will be asked at the end of the lecture. So it is my pleasure to introduce our speaker today, Dr. Brad Imany, who I've actually known for up to a decade. We just discovered this over lunch. I actually really got to know Brad in 2020 at my previous job as a program director at the National Science Foundation when I started a program, an Emerging Frontiers in Research Innovation program on brain-inspired dynamics for energy-efficient circuits in artificial intelligence. And Brad was extremely generous with his perspective and knowledge. Brad is a distinguished member of technical staff in the Center for Computing at Sandia National Lab. He has led several research projects that leverages computational neuroscience to advance artificial intelligence and in using neuromorphic computing platforms for future energy-efficient scientific computing. He recently led a multi-institution Department of Energy Office of Science microelectronics co-design project on novel probabilistic neuromorphic computing called coin flips. In 2023, Brad was the recipient of the prestigious Misha Mahawat Prize for Neuromorphic Engineering, and he was also a National Academy of Engineering U.S. Frontiers of Engineering invitee since 2017. Before joining the Department of Energy, Brad studied with Rusty Gage at the Salk Institute as a computational neuroscientist in Neurogenesis. You will hear a little bit of that research today. And he will tell us about how neuromorphic computing can help us understand the brain. Brad? Let's welcome our speaker.
Well, thank you, Grace, and thank you, everyone, for this amazing opportunity to come here and speak with you today. It's quite an honor looking at the past presenters of this lecture series, and I think it's an exciting time to be able to come and share some of the things that are going on in my field, kind of the neuromorphic computing field, which is a new field for me. As Grace mentioned, my original work as a neuroscientist was looking at adult neurogenesis, which I'll tell a little bit about to start. And over the last decade or so, I've been focused on the computing side, and somewhat surprisingly, neuromorphic computing hasn't had a lot to do with neuroscience historically, but that's changing, and I believe that we have a real opportunity, if we choose to take advantage of it, to bring these fields closer together and help us, gain new insights about how the brain works. So I'm going to start with a kind of a metaphor, if you will, or an inspiration, if you will, and looking at how modeling and simulation has had a tremendous impact on our society as a whole. This is Hurricane Milton that, if you're familiar with the news, and most people are, hit Tampa a couple weeks ago. And what's amazing about this is we're all familiar with these sort of cones of uncertainty where these storm tracks may come. And this is a case where the average person is seeing modeling and simulation impact their lives in real time. Right? Day to day, we actually get a new forecast of where this storm may go. And this was an unusual one, of course. It went from west to east. That's not what hurricanes normally do in that part of the country. But what's I think even more amazing is that this storm did almost exactly what the models predicted. Now, that doesn't always happen, but it's happening more and more often. And it's because our ability to simulate these systems, this sort of complex weather, incredibly complex weather systems, has gotten better and better every year. And part of the reason why is if you look at these sort of supercomputers that NOAA has, they run, I think this is in Virginia or Arizona, these large-scale, giant, high-performance computing systems, in effect, can be deployed, saving untold number of lives and a tremendous economic impact. And the question is, why are we not able to do this today for mental health and neurological disorders, right? This is kind of a challenge, right? Motivating sort of thought. Wouldn't it be nice if we could make that same sort of prediction? Of course, other people are thinking about this as well, but I wanted to come at it from a different perspective, which really is looking from the bottom up, right? As someone who modeled biological process, kind of at high level of granularity, at least at the time, how do we go from this level of understanding into this sort of system understanding of how the brain works and how diseases may be understood and be treated? So if we want to start building these sort of high fidelity models of neural processes, neural diseases, specking out treatments and so forth, what do we need to do? So my talk, I want to kind of back up, kind of tell you where I'm coming from on this, about my original work in looking at adult neurogenesis in Rusty Gage's lab at the Salk Institute, and then how my work in that area, as well as the broader community, started kind of help shape a path forward, how we're going to potentially, what we need to do to get there. And then I want to talk about this field of neuromorphic computing, which is really a new thing. I imagine for many of you is probably a new concept. It's something that is really an exciting area that's really emerging on the scene right now. And talk about how that may actually have real unpredicted insights into how neural computation may be occurring. So to start, I want to go back to adult neurogenesis. This is a process that is quite, I still find very exciting. The idea that right now, all of us in this room, all of us listening online, are experiencing the addition of new neurons into our hippocampus. Not a ton of them, mind you, but they are happening, right? Development does not end when you're 5 years old, the way that we were taught, growing up, right? There are new neurons in the adult brain. Now, this is a localized process, as you see here. It's kind of highlighted. It's in the dentate gyres of the hippocampus. On the bottom right, that's an image from Chunmei Zhao, who was a postdoc when I was a grad student, who took these beautiful pictures of these GFP-positive neurons. The green cells are the newborn cells, about a month old in a mouse. And it's particularly a small population of granule cells, which are the dominant cell population in the dentate, which are the sort of pink cells you see there. So as I mentioned, this was kind of one of these, people love to use this as an example of one of these dogmas that was overturned. We now know that neurogenesis does exist, even though we were told it does not. But it's not a ton of cells, as they said, right? In humans, it may be, at any given time, having a few thousand neurons that are in immature critical state. Still to be determined. This is very difficult to measure in humans for obvious reasons. We do know, though, that in rodents, this has a real impact on memory formation. It seems to be clearly important for cognition. And we also know that it is highly correlated with a lot of things that are clinically of a lot of interest. Things like epilepsy, depression, stress, aging. All of those seem to have some interaction with neurogenesis, and at the same time, some things like SSRIs appear to interact and exercise. Other sort of things that are seen as treatments also do have effects on neurogenesis as well. So I'm not going to go into the full depth. This is a whole field. It's been kind of a community of people studying this, including here at NIH, over the last couple of decades. But what I want to talk about a little bit is the computational impact of this, right? So what's convenient about this process is that we were adding new neurons. Apparently, the addition of new neurons is occurring in a brain region that is relatively well studied from a computational point of view. Dentate gyrus, of course, is where LTP, I think, was originally seen in vivo in mammals. And what people were observing while I was a grad student, a lot of the field was kind of resonating around this idea, as you see on the left here, that these new neurons, as they come in over the course in a mouse of multiple weeks of development, that they go through these periods of kind of increased excitability and increased ability to learn. They start off kind of quiet. They're stem cells. They're not really wired into anything. become kind of hyperactive. They're like, hyperactive little children, as you see on the figure on the right. And the mature cells, they eventually mature and they get quiet and they only respond to what they're supposed to. So this sort of model of computation was appealing. It was kind of attractive because what we have is a case where there's this classic coding scheme in the dentate gyres where each neuron responds, in effect, almost like a grandmother cell. It's not quite a grandmother cell, but it responds highly selectively to a very small set of things. But the young neurons, for a transient period of time, might be quite receptive to respond to new things, and in that process, learn to respond to those later. They basically sample lots of possibilities and then become an expert in one or two things. So this was compelling and interesting from a computational point of view because it makes you start thinking, well, what does it mean to build a computing system, an algorithm, if you will, around this? This is before AI really took off, of course. So when we started thinking about what this meant, I won't go into the modeling itself in depth, but the conclusion that we really had was that the dentate gyrus has been known and believed for many years to be important in something called pattern separation, the ability to discriminate between relatively similar things. And you can see this from the sort of animal behavior side where an animal's context fear conditioning needs to discriminate between two similar environments. Physiology, where different populations of neurons respond to similar inputs, and so forth. But what was complicated, and I still find complicated, is that if the dentate gyrus is really good at pattern separation by construction, and there are anatomical reasons to think that would be the case, why would you need to have this complicated process of neurogenesis to do that? You don't need neurogenesis to pattern separate. And then, especially given that the rest of the brain has shut off neurogenesis. I mean, the vast majority of the brain does not have new neurons coming in throughout life. So clearly something more has to be happening. But we just didn't have the tools to understand it. So really the challenge that I had, at the time, and this is I was finishing up my PhD and some time as a postdoc, and trying to understand if I have this sort of conceptual model of what new neurons may be doing, but I want to get to the brain. I want to, we're in it to treat diseases and, improve cognition with aging and overcome some of the effects of mental health on cognition and so forth. And we know neurogenesis is likely involved, but how do we connect those dots? We need to expand in some direction. And the two obvious ways, looking at it from my perspective as a computational person, was maybe it's a challenge with modeling, that we simply don't have a big enough model. We don't have a realistic sized model. At this time, this is, again, before AI, before GPUs that could run billions of neuron models. Maybe I just need a bigger model. But even if you look at this, it's a lot of neurons in our brains. I mean, even compared to today's computing capabilities, we have a lot of synapses, a lot of times, milliseconds in a month. And then there's the other issue, which is on the right, which is that the biological complexity is tremendous. There's so much we don't know. And one of the examples of what we didn't know that, of course, is going to be important if you're talking about neurological conditions and mental health is that, it's not just GABA and glutamate and you're done, right? There's a lot of immense complexity in neuromodulatory systems that's really important. And so when I was first thinking about where I want to go after my time at UCSD and Salk, I was motivated and kind of inspired to think, can we start putting the complexity of things like serotonin and dopamine, norepinephrine, these modulatory systems that are known to be critical in cognition and in affect and mood, that are often implicated in disorders. And we know from electrophysiology work that these are important when it comes to things like the behavior of granule cells and the interneuron populations. So along with a postdoc, Yangling Mu, who was in Rusty's lab at the time, who left and went to start her own position in China, I did some modeling work where we put dopamine, and it's a very abstracted model, so the details are basically what you see in the top of the chart, a very simplified model of dopamine based on the physiology results she was seeing. where we observed that when you put this into this sort of pattern separation model that we had, you see this sort of temporal asynchrony that happens. So basically, if I have some sort of behavioral model, I'm adding new neurons in there, and then I have a dopamine event, the things that happened before the dopamine event were encoded differently than the things that happened after the dopamine event. Now, if you're familiar with AI and where some of the AI techniques have gone in recent years, this is actually not all that different from reinforcement learning, which of course is, dopamine has been implicated in, and temporal difference learning. And these are some fairly fundamental building blocks of AI. So we further went in, and this was work that Yang Ling's lab did, where they did behavioral experiments and physiology experiments where you give a mouse condensed milk, and you can observe that, in effect, after a dopamine event, or presumably condensed milk is the equivalent of a dopamine event, it kind of induces a reward response, the learning is suppressed, not all that different than what the model suggested. And then this went in and looked at context fear conditioning and some other things. I'm not going to, for the sake of time, not show all those results. And I think this was exciting because it showed that, yes, there's a real reason to include the complexity of modulatory systems in these models. But the problem is, this is one modulator with a lot of work that was done by a number of experimentalists who are on this paper. It was in PNAS at the time. And and we were able to kind of nail down one experiment and kind of one little model result. And it's like there's so much more. Right? It's just, this was like chipping off a tiny little bit and hoping that, we just have to do this forever, right? And so that was a little underwhelming. I mean, it was exciting. I love the result. But it's like, is this really a path forward to kind of, transmitter by transmitter, cell type by cell type, chip away at what's going on? So as a way of kind of framing this, many of you are probably familiar with Pat Churchland and Terry Sinowski's. They had a book in 1988. About 10 years ago, they had this follow-up paper looking at the time scales of experimental modalities, spatial scales, and of course this differs tremendously whether you're doing single spikes in physiology or fMRI imaging. This same thing happens with plasticity. Neurogenesis is occurring, of course, I mentioned weeks and month scales and circuit scales, but the modulators are in whole different time scales. So in a sense, you start looking at this, and the time scales, the spatial scales, the complexity scales becomes a little daunting. But we have giant computers. Shouldn't that be enough? So for this reason, kind of looking at that complexity and thinking, okay, I'm not going to solve the problem one system at a time, especially as a modeler. I love working with experimentalists, but, that's not necessarily the best way to start a career, is where you're dependent on a bunch of people to give you data. So I went to Sandia National Labs. Now, if you're not familiar with the DOE laboratories, they generally don't have a lot of neuroscience. That's beginning to change. At the time, they didn't have much at all. What they do have are really, really big computers. In fact, the biggest computers in the world right now are at Oak Ridge National Labs and Argonne National Lab. And we're hitting 10 to the 18th flops. And at the time, Sandia, when this is about 13 or 14 years ago, Sandia had one of the biggest supercomputers that was available to Sandia's staff to run experiments on. So I went and I said, okay, let's go to realistic scale dentate gyrus simulations. Maybe that's going to solve this problem. And, again, I didn't show the model that I did originally, but it was granule cells with some intrarenal cortex inputs. I said, well, let's look at all the different interneuron populations. Of course, there are many more than this, but this is more than you see in most models. 6 different interneuron populations, different types of intrarenal cells, grid cells, and non-grid cell inputs. And then we'll look at some sophisticated functions, the pattern separation function I meant before, and the ability to encode information. What we observed, which I believe was the first time we ever saw that this had really been shown, although intuitively it makes some sense, is that there is a real value of going to large scale. So again, this is 15 years ago. 25,000 neuron model was still at the time one of the biggest models around. But that's only 10% the size of a mouse dentate gyrus. For context, the mouse is at 300,000 cells, which you see on the top right. and a human is about 10 million cells. So if we scaled up on the supercomputer, as we kind of increase the size of the network, we see, you can see on the bottom right, that basically the activity of the overall network decreases. And this is, there are real reasons for this. If you look at, there's a difference between a neuron that has 10,000 synapses and a neuron that has 100 synapses or 1,000 synapses. Scaling is not linear when you're talking about things that are highly connected in a complex graph. So a lot of the models that we had, and this kind of, is like a warning sign, right? A lot of models that look at brain regions and say, I have a 1,000 neuron model of whatever region, unless your region actually is 1,000 neurons, like a locus coeruleus model, say, you're probably getting a biased result because there's a real complications in scaling. What was interesting, though, was that when we add new neurons to this, we saw that the overall scaling effect didn't change for the mature cells, but the young cells didn't care. In a weird little sense, the young neurons, because they already are, they already have fewer connections than the mature cells do. They're kind of small network neurons to begin with. So they didn't care about the size of the model, but the mature cells did. And we looked at pattern separation and information coding. We saw that it was improved with neurons. So this was exciting for one reason, which I didn't really go into, which is that, as I mentioned, human neurogenesis is thought to be at a low level, much lower than mice relatively, but this would suggest that as you go to larger scales, you don't need as many new neurons. So that was exciting. But what wasn't exciting is after doing all that work, and that took a while to build this model and to run these things, I realized that we didn't really learn much. And this was a bit underwhelming at the time because we were looking, and I realized it was because we were looking for pattern separation and we found pattern separation. And this is a challenge that often happens in neuroscience, and I think it's a big challenge in systems and computational neuroscience in particular, which is that the system, things are so complicated that we often construct our experiments to look for a particular hypothesis. But since the systems are really complicated, it's pretty easy to find validation. So confirmation bias is real. It certainly is in the case of these simulations. Things like finding decorrelations in data is not hard. So I was a little, this was a little discouraging. And in a very real way, it led to me kind of dialing back a little bit on this bottom-up simulation effort. And part of the reason was that, when you start sitting there and ask what you really need lots of data. We didn't have that much data at the time. Getting it about dentate, but it's not just the dentate. Everything else matters too. The time scales, as I mentioned, are a big deal. And we have this big growing issue that I didn't know what to look for when it came to the computational models. Like, what is the task I want to do? It's like context fear conditioning, which we saw effects of in mouse, involves a lot of systems, a lot more than the dentate. So how do we map these? So as I mentioned, I think we had these sort of hard lessons about, you can model something big and complicated or small and complicated or big and simple. We need better behavioral tasks. I was a little bit at a loss, and this is me about 10 years ago. Well, what happened about 10 years ago? Well, I think everyone here is very familiar with one thing that happened was that there was a major investment in the BRAIN initiative that wanted to, I would say, wonderfully said, let's get to this biological complexity challenge, right? We don't know enough. I just showed you why, from one example, we don't know enough about aspects of the brain to make some deeper insights. And the BRAIN initiative faced this head on. The Human Brain Project, I think, did the same thing at a different scale and had a different emphasis, of course, but it was happening around the same time. And another thing happened around this time, which is that artificial intelligence exploded. Now, AI is not biological intelligence. That's why it's artificial, not biological. And what I think was, if you look at it and you look at what happened, in some ways it did the opposite thing. It stripped away whatever biological complexity remained in things like neural networks and focused on let's make them as big as possible and as simple as possible. But at the same time, we're doing both, and maybe these things can combine. And one of the ways they combine is what we would call neuromorphic computing. that actually I believe is a path by which we can get complexity and scale together. And this is something that, it's not in lieu of these other things. We absolutely have to have this sort of focus on biological complexity. And the AI field, of course, is not going anywhere. Artificial neural networks are the world we live in now. How do we take advantage of this? And I believe we can start pushing up to the top right of this. So what is neuromorphic computing? I've just thrown that term around a few times. Neuromorphic computing as a field has existed since the 1980s. Carver Mead is a electrical engineer, very critical at the early stages of building kind of integrated circuits. And towards the end of his research career, he and a grad student, Misha Mahwald, decided to actually begin to emulate parts of neural systems, as understood in the 80s, in silicon. And that sort of, is Misha's vision originally, where, let's put what we know about different sensory systems and build silicon chips out of it that capture that. And that field kind of took off. There's a kind of a community of researchers. They focused a lot on things like event-based sensors that have led to things like artificial retinas and cochleas and so forth, and event-based cameras. And in about 10 years ago, that field was kind of going along, looking at analog, which is non-digital silicon electronics. About 10 years ago, there was a renewal of interest from the more computer science community, partly driven by some of the stuff going on in AI, of looking towards large-scale systems. And it's basically because the brain's really efficient at computing, and our computers today are not very efficient at computing, in terms of energy, I should say. So there are two broad classes of neuromorphic computing. I'm not going to go into the gory details here. We focus at Sandia largely on digital CMOS technology. So this is things that are the underlying fabrication technology of a Intel Loihi chip, which is what you're seeing unboxed here on the bottom right at Sandia. Those chips are the same materials and fabrication that's in your cell phone, your laptop, whatever. But the architecture, the computer architecture, is not von Neumann. It's not programmable the way you would program a normal computer. It doesn't naturally read Python. And it's an architecture, rather, that looks more like the brain. You program circuits of neurons. Now, the other alternative is analog systems. I'm not going to go into detail. We do some of this work at Sandia. It's further out. It's more capturing the biophysics. It has a longer path towards scaling. It's probably the future, but I say I'm going to focus more on the digital CMOS technology. So this is more of a computer architecture-oriented slide. There's lists of things from the brain translated into electrical engineering or computer engineering concepts like spiking, complex networks, the ability of synapses and neurons to exist in the same physical location, learning. All of these things are what these hardware platforms try to capture. And it's a rapidly evolving field. I think the full details of it, they will continue to change over the course of the coming years and decades. But right now, we can get 1 billion neurons onto each of these little chips you see on the top right. Or sorry, 1 million neurons in each of these little chips. We can get billions of neurons in a platform which is about the size of this large microwave here. And we have one of these at Sandia. We believe it's the world's largest neuromorphic system that's in our lab there. Each of these chips is 1 watt of power. That's about 100th of a CPU that might be in your laptop, and maybe about 1,000th of a production level GPU, give or take. So I'm going to talk about neuromorphic. I'm going to give 3 sort of vignettes for this last part of the talk, where I want to show what a neuromorphic view of computation can tell us about the brain. Given this sort of neurogenesis-inspired vision I had before, and kind of where those challenges are. The obvious one is that we can maybe get to things at scale, and that's what I'll start with. The second one is that I believe it provides us some interesting constraints of how we're going to analyze data that comes out of larger and larger neural systems, because in some level, if we have millions of neurons on our chip, we're going to need to analyze that data if we start simulating it. And then the final thing, which is a little bit more out there, I will admit, is that we might be able to actually uncover some new directions to think about how the brain actually is working by looking at what these chips are good at. So I'm going to start off by talking a little bit about our ability to simulate neural systems at scale. So of course, the dream is if I had a connectome of a brain, eventually a human brain, but a connectome of whatever brain you care about, could I actually model that connectome relatively easily on a neuromorphic chip? And now connectomes, of course, are lots and lots of neurons, lots and lots of synapses. We generally don't have them. Well, as many of you know, about a month ago, or less than a month ago, the Flywire team at Sebastian Sung and colleagues, largely based out of Princeton, work that I believe the Brain Initiative helped fund a large part of. The Flywire team published a series of 9 papers. These are new. I've made my way through a couple of them. I will admit I haven't read every single detail yet. And part of what they had in this paper was a full connectome of a Drosophila brain. And this is pretty exciting because we've had a connectome for a long time of C. elegans, but 307 or so neurons that are mostly analog is quite different from a model system that is in the hundreds of thousands of neurons and can do some very sophisticated behaviors. So one of the papers that was published here is a model of sensory motor processing, looking at kind of, I think, an olfactory response that is a model of the full brain, right? So they have the connectome and they implemented a model of it. So we asked the question. I'm going to say we asked the question, but I didn't do it because I've been traveling, but a couple of my colleagues here at Sandia, William Severa and Felix Wang and Brett Thielman, they decided just kind of like, well, we have these chips, we have this connectome, they published everything with it. We've never really talked to the Flywire people, they don't know we did this. But they published it, it's there, and we said, let's take your model or take that model and stick it through a couple of our tools, Fugu, which is a spiking programming tool, and Stax, which is a large-scale simulation tool, and map it onto Loihi. And we did, or they did. I'll give Felix and William most of the credit here. But we actually were able to take this model off the shelf, literally off the shelf, download it from GitLab and just stick it onto Loihi. And it took a little bit of work. I'll admit that it wasn't an instantaneous thing. But what we see are dynamics that are coming off of these chips, the neurons in the chips that are largely analogous to what we should see. Now, this is, quite literally hot off the presses, right? This is, we haven't gone through and validated every single thing yet. There's some design consideration we had to do. But this says that these chips coming off of things like Intel's Loihi and when Spinnaker, SpinCloud, Spinnaker 2 chips, these things can run biological models, and it's a question of engineering now to how we actually fit the biology data that we're producing, or you all are producing, onto the chips that are being produced. So I think this is pretty exciting. I think this was quite exciting. I think it shows that neuromorphic computing has a real value. Now this is not a huge network nowadays for, you can put on a GPU. I don't think there's we don't have to have neuromorphic to study fruit fly brains, at least at this level. We may find value there. But as we scale to larger systems, I think we will. And this says there's a path. All right, so the second thing. So as you can see from this last plot, there are a lot of neurons. This is only plotting 100 of the 100,000 neurons. A system that when we simulate actual cortical neurons, we did one in our supercomputers at over 26 million neurons using the Stax tool. So 26 million neurons coming off, modeling a loose cortical recurrent connections or whatever. You just get a raster plot. We call this a raster plot in Comp Neuro and Systems Neuro, where you have over time lots of neurons and they're firing and not firing, right? It looks like noise. So we've effectively created a new big data problem. So one of the things that was kind of an interesting thought when we put these big neural simulations onto these supercomputers, and this was on the Oak Ridge Summit machine, we saw that we had this temptation, which we all have, was, well, I'm simulating every neuron, let's record from every neuron, which creates that big data problem. But when you do the neuromorphic system, if you record from every neuron on a neuromorphic simulation, for reasons that aren't worth getting into, you lose all the advantage of being a neuromorphic. If you have to like read out gigabytes or terabytes of data, right? So input-output is expensive. So ideally, you only want to observe a subset of neural activity, which is exactly what we deal with when we're looking at the brain. Like, I can't imagine a future where we're going to record the spiking activity of every neuron of a patient's brain, right? We're always going to be looking at a subset. So what do we do if we only can look at a subset? I mean, how do we go from spikes to something a little more abstracted? So this is work of my postdoc, Brad Thielman, who came from the birdsong community. He was in Tim Gettner's lab at UCSD. And he had this realization that as we're looking at this sort of large-scale data, we tend to do what people trained in serial computing programming do, is think about chunks of time, you know, what spiking events happen at different periods of time, and then that sort of pattern of time kind of progresses forward, and I get some trajectory through time and space. And what Brad's realization was is that there's no reason that the brain needs to be, you know, the brain's not chunking things up in time. Right? Time is kind of an artificial construct because that's how we measure things, that's how we simulate things. But these neurons are just they're firing and behaving asynchronously based on their inputs, maybe a little bit of loose global information. So what there's been a lot of work looking at causal relationships between neurons in these sort of raster plots. And what Brad realized is that if I'm doing a simulation, I have the connectome, right? I simulated it. I know what connects to what. And if you combine that sort of functional causality measures that people have been doing for a few years, with structural constraints, you get something that's analogous to a spike timing dependent plasticity rule. Won't go into the detail. And coming out the back end, we get something that we call graphical neural activity threads, or NATs. We like acronyms. We're national lab. Government likes acronyms. So our NATs actually decompose this sort of complex raster into these series of overlapping populations of causally linked neuron patterns. And what's really great, and this is a paper we have on arXiv, we're writing it up for, in a more formal way right now, is that you see these same sort of correlated patterns can exist at several different times in the system. It's not just like a one-off thing, but rather you're seeing like ensembles that are spatial temporal that will repeat themselves over time. Moreover, if I give a certain set of inputs, I will see certain patterns reliably reactivate themselves. Maybe not every time. There's stochasticity in the model, and so they're kind of random when they pop up. But it seems like this is actually a fairly scalable approach to abstract away from the sort of like neuron 1 fired at time 1, neuron 2 fired at time 5, or whatever thing, right? This is like zooming out a little bit, maybe make something that's a little bit more useful from a computing point of view, and potentially from a monitoring point of view. So this is early days. We just got a CRCNS project through DOE that's working with Tim Gettner at UCSD and Joel Rosenfeld at USF to look at this in a little more detail with some real data. But we're pretty excited. All right, grab some water. So the final step that I'd like to talk about briefly is this idea that we might be able to get a deeper understanding about how the brain is functioning from looking at what these neuromorphic systems are good at. So our hypothesis here, and I will say that I think this is somewhat of a radical hypothesis. It certainly is in the neuromorphic field, is that neuromorphic computing potentially can help us understand things that the brain is actually good at by virtue of having been constructed of the same sort of architectures and the same sort of design principles that we believe the brain has. Can, in a sense, neuromorphic computing help us get something better than just saying pattern separation, but something a little more computationally rich and useful? I want to contrast this by saying that neuromorphic computing, I believe, is necessary for this compared to just saying, I can use AI to do this. And part of the reason is that artificial intelligence, while as powerful as it is not constrained by physical space or by energy constraints. We know this because we can look at the energy costs of today's AI algorithms in production. It is always a possibility that if I need a neural network to do something, I throw more data and more layers, and I can get it to work. That doesn't really tell us anything about how the brain works, because the brain can't say, I need to go learn German, I'm going there next year, why don't I add a few more layers to my cortex? That's not an option for us. Our brains have existed with constraints. So I don't think the fact that neural networks are successful at doing cognitive things is as useful as we might wish they were. I think we have to include the constraint of efficiency. And the reason is because it's not the amount of work that an algorithm requires, but it's the amount of work under a fixed sort of constraint. Our brains have to deal with that, and these neuromorphic chips have to deal with that. I have a concrete power, they're low power, they're designed to be like the brain, but I can only fit so much on them. So what can I do with that? So if you believe that the neuromorphic chips that we're developing have at least an overlapping set of constraints that the brain has, we believe that we might be able to actually make some insights about what we can do. So very quickly, this is what a neuromorphic system looks like. If you're familiar with a leaking integrating fire neuron, it's a fairly simple model of a spiking neuron. Most neuromorphic chips today have hundreds of these leaking integrating fire neurons on an individual compute core, and then a chip has a few hundred cores, and then I can wire up a bunch of chips together. So a nice hierarchy. But what I'm programming are a bunch of neurons talking to other neurons through synapses, same as our brain does. There are two functions in kind of the remaining 10 minutes here that I want to show that we have discovered neuromorphic computing is highly efficient at. Things that we did not expect it to be successful at. And one of our, kind of my day job at Sandia is to look at neuromorphic hardware and say, what can this do for computing in general? Because Sandia as a whole is very worried about, and DOE as a whole, very worried about the energy costs associated with computing today. So what can neuromorphic computing do efficiently? So we've discovered two things. One is that problems that require widespread sampling, which you might, our brains, of course, do this synaptic sampling, and problems that are spatially distributed, solving kind of getting physical solutions to problems that have spatial constraints. So the first thing was done primarily by William Severa, who did the flywire mapping, and Darby Smith, who was a postdoc of mine, who's now staff at Sandia. Both these guys used to be postdocs, and now they're staff at Sandia. They're mathematicians. Mathematicians are fantastic to work with. I highly recommend it. So I mentioned earlier the hurricane prediction. This is a different hurricane. But of course, there are a lot of problems in the world that we care about that rely on uncertainty to understand. Our computers today are not very good at dealing with uncertainty. Flipping a transistor is a binary, discrete, deterministic event. So what can we model things, probabilistic problems, on neuromorphic hardware? And this was something that people would never have guessed was possible. But this is what, about 5 years ago, we really realized that the paper came out in 2022, where we saw an actual neuromorphic scaling advantage for modeling random walks. And if you don't know what a random walk is, imagine the stock price kind of jittering up and down. It may go up over time. It's going to go up over time, but the path it takes to get there is stochastic. It's not predetermined. So I won't go into how the model works, other than to say we were able to wire up populations of neurons in a way that represent the state space over which a random walk has to occur. A Monte Carlo process kind of evolving over time would basically be akin to a spike moving over this mesh of neurons. Every random walker in my model is a different spike moving over my chip. That's a radically different way of thinking about Monte Carlo simulations and computing, and it works really well on neuromorphic hardware. What essentially happens, my 11-year-old did this in Blender, which amazes me. I can't do this in Blender. But every neuron in the system is behaving independently of every other neuron, or every mesh point is behaving independently of every other mesh point. And so the random walks just kind of go in full perfect parallelism. And this turns out to be a very huge energy savings. We saw this in our benchmarking at the time, where you see that the number of walker updates per joule, so we're measuring energy, not power here, is orders of magnitude better than what you see on a CPU and GPU on neuromorphic hardware. So it went a little bit slower on some of the original neuromorphic systems. Now these new systems are able to go faster, but we still think the power advantage is still there. So this is saying things like probabilistic sampling, Monte Carlo random walk, possibly Bayesian inference, things like this. Real problems that the world really needs to deal with are very nice fits for neuromorphic hardware. Of course, our brains similarly have to deal with uncertainty. So then the second area is also work of my current postdoc, Brad Thielman, who did the NAT stuff, where we're looking at spatially distributed physics problems. So if you do computational physics, many problems can be solved, kind of described as a partial differential equation that looks generally like this. And the way you would do this classically is using something called finite element methods. Just as if you're not familiar with the process, it's basically trying to understand some physical dynamics like heat or whatever over some surface. Every point on that sort of mesh, I slew up my area. I think I'm modeling over a mesh or something. And at every point, I basically compute the physics at that point based on the neighboring points. And this is the gold standard of how you do things like turbulence modeling or heat diffusion modeling and so forth. There's a lot of work these days in using scientific machine learning to approximate this, but the real question is can you actually map this onto neuromorphic chips? Again, no one anywhere in the neuromorphic field would think this was possible. But we wanted to, based on that probabilistic result we had before, can you actually use some sort of sampling approach to do this? And what Brad, who had a realization from his neuroscience background, realized is that you can actually, if you have these sort of cortical models where you have clusters of neurons that are excitatory and inhibitory that are, kind of locally optimizing themselves to represent something, well, you can wire those up in a mesh, not unlike what the finite element mesh was I showed. And mathematically, you can actually make those relationships explicit, such that I can take the physics model that a computational physicist cares about, AX equals B, you want to solve for X. I can take that A matrix, and I can represent it as the connections between these mesh points, which you can kind of see here. Again, the details, it can get pretty complicated, but it's not requiring any dynamics or math that is not something that neuroscientists have been looking at for years. In fact, it's rather nicely parallel to things that have been in like more control perspectives of how cortical circuits may work. And it scales very nicely on things like Loihi 2, which we've been doing. And if I hid the labels in the bottom there, you wouldn't be able to tell me which one was the neuromorphic solution and which one's the ground truth solution. And this is something that, again, everyone thinks that an AI solution or a neuromorphic solution has to be some coarse approximation of what we can do with, fancy calculators and deterministic computing. It doesn't have to be. It turns out that these neuromorphic chips, using brain-inspired algorithms, using neurons as the underlying compute units, can solve these problems at levels of accuracy and precision that a computational physicist may really care about. And if you're like a cortical systems neuroscientist and you look at the sort of raster plot here coming off of the model, this could easily be data coming off of some sort of multi-electrode array or something like that. So this was pretty exciting because it kind of tells us that these are physics problems. These aren't biology problems, right? But our neuromorphic hardware seems to be really, really good at doing this. And if you stop and think for a second, well, what are these problems really doing? They're trying to understand what the physical world is based on some perhaps observations, but definitely some sort of prior model, and want to make predictions of what's going to happen. Well, we have to do that. Our brains have to do that, and that's a real problem that our brains have to solve and have trouble solving when things start to go wrong with them. And likewise, the ability to make decisions often requires sampling a bunch of potential outcomes, which is something that means that our brains really probably need to do some sort of sampling process, some sort of probabilistic reasoning. And of course, Bayesian reasoning is something we've long thought the brain is doing, but I don't think we've thought of it by doing this sort of parallel Monte Carlo sampling in the way that we've seen neural circuits are actually quite efficient at doing. So what I would say, and I maybe challenge the neuroscience community here, is that instead of thinking, oh, I think the brain must do this because I study cognition and this seems important, what's the formula for this, how does that map to the brain? Maybe we can go the other way around. Circuits of neurons are good at doing certain things. What are those things? And if we can understand what those things are, then can we look into the brain and see if that's showing up somewhere? And indeed, there are cases where there was a beautiful paper out of Europe a few years ago looking at random walk trajectories during hippocampal replay. So the replay events might actually be a Brownian motion sampling. And then there's some very nice predictive control models that are actually quite similar to some of the models here. So to kind of wrap up, we have this challenge where we're looking at the complexity of the brain, the timescales and the size of the brain, and this kind of challenge of computation that we're faced with. And we don't necessarily know what to do. And what I believe is that neuromorphic computing is not the answer here, right? We absolutely have to get more data, and we have to understand more. We need connectomes, but we need more than connectomes. But I also believe that having this more brain-inspired computing constraint, kind of getting away from this idea that we can do everything by just having enough layers of neurons on it, but rather constrained to the physics of this emerging hardware, may actually give us a path to do this. And I'm supposed to say neurorealism, but I believe that we can then, as we learn more from the biology side, influence neuromorphic computing and the trajectory it goes. And I had a question at lunch about, how a neuromodulator is going to end up on neuromorphic computing. Well, certainly no one is thinking about that on the neuromorphic hardware side at a deep level because we don't know enough about neuromodulators like serotonin to tell them what they need, but I think it's not unreasonable. The field is still young enough that we can influence it. So with that, I would be happy to take any questions. I would like to first off thank, this is a large group of people I've worked with over the years, fantastic collaborators, experimental and computational, mathematicians, computer scientists, all sorts of people. So thank you very much.
Well, I certainly encourage anyone here to use the mics. We have one question online to get things started, so the rest of you can use your brain and or computer to think of questions. So this is a good one. Ready? Mistakes are inherent property of neural networks-based biological and artificial systems. Can neurosymbolic-based AI help to decrease number of mistakes made by AI?
I think it's possible. So neurosymbolic processes, it's a little bit different than how I was describing things, but the idea is that we know that there are two types of AI where you have kind of classic symbolic models that experts might construct, and then these neural networks that are quite powerful, and you combine the two. And the hope is that this might provide explainability and more accurate modeling. And what I might twist the question a bit and say that our neural circuits make mistakes for a reason many times. That's how we learn, right? Our brains are lifelong computers. They're not instantaneous computers. We develop our neural networks as, we have our training period, and then we validate them, we deploy them, and ideally they're not changing much. But our brains, of course, have to change continuously. They have to adapt to both the changes internal as well as external. And part of the way they adapt is through these mistakes. So I would probably say it's not about reducing errors. I mean, I know that's how we tend to quantify AI performance, but rather making sure that when a system explores possible alternative scenarios, right? Sampling, if you will, the way I described it, that actually doesn't throw things completely awry. It doesn't, crash the car, if you will, if you're like a, self-driving car. Of course you don't want that. But you want to explore, you have to make mistakes. That is what cognition is. So I think a little bit of exploration and mistakes is good for a system. Hope that answers the question.
Oh, please. Oh, go for it.
Thanks, Brad. Really, really nice talk. So you showed at some point a simulation you had implemented from the Flywire data. And one thing that was impressive is that it looked like a really large number of neurons, right? The fly data is a really large number. So I'm interested in trying to, I'm wondering what kind of architecture that uses. Are you like implementing the sort of analog integration directly on chip? And then I guess more broadly, I'm sort of interested in where you see the large scale of neuromorphic computing going, because one of the, I don't know, people say that our brain works better than ChatGPT on like like a hamburger every few hours, and that's a lot less power than AI uses. So one of the future things is, one of the directions we're thinking about in the future is being able to do really large scale computation in this analog format using relatively low power. And so I'm wondering where you think like the scale of this system is going, and are you going to be able to scale it up to really, really large numbers of units, and how will that happen?
Yeah. All right. So if I forget the second question, remind me, but I think I'll get it. So quickly on the Flywire work. So this is, like I said, it's hot off the press. We haven't fully, tested everything on it. The paper just came out a few weeks ago. But what was nice is the paper that we are based on is not just the connectome. It's actually a paper that described a computational model from the connectome. And that model was written in Brian, which is a spiking neural network modeling tool, which effectively is doing leaky, integrate, and fire neurons. They're a little bit more complex, but not tremendously so. So we just, what was nice is that model we could basically take directly from the code and translate it onto what the core digital neural models are that are implemented by the Loihi 2 chip. So they're not analog. It's fully digital. It's basically, the way that chip works is that it has 100 some odd cores that are each capable of modeling about 1,000 neurons each. In practice, you get maybe a few hundred neurons each on each core. So it was just more or less a jigsaw puzzle thing, right? We wanted to take that model that had 125,000 neurons, split it up into pieces, and distribute that over the Loihi 2 system that we had. So that wasn't, what's nice is, we didn't do the work. I don't want to take credit for any of that. That was all the work of the Flywire team by making that model. We just showed that you can port that to Neuromorphic. The second question is kind of this, how is this going to scale and where is it going to go? And I think it depends a little bit on if you want to do more of this sort of like brain simulation stuff, or if you want to go this AI route. So I think for the brain simulation side, we'll have a path there. There's some engineering constraints. The number of synapses per neuron is bounded by the local memory available per core. So if you think about something like a locus coeruleus norepinephrine neuron, it might contact 1 billion neurons in the brain, right? And that sort of graph structure, we're going to have to figure out how to map that to physical hardware. But that's just going to take some work. I think it's possible, and I think we can probably get to realistic sizes. Like we said, we have a 1 billion neuron system today that we're trying to figure out how to build things up on. So the limitation, there are limitations, but there's nothing fundamental limiting us there. When it comes to something like large language models, I would say that the underlying structure of those models is not very biological. It might include things that are neurons and weights, but those models in many ways are not good fits for neuromorphic hardware. I would say, people are trying to do this. I'm cheering them on. I'm not incredibly optimistic that we're going to put ChatGPT onto Loihi 2 anytime soon. What I think we might be able to do is seeing what these chips are really good at might help constrain us to think about what a different formulation of a large language model should be. A different formulation of a large language model should be, and that might help us re-describe it in a way that is energy efficient, which I think is a very interesting scientific question, but I don't have an immediate answer.
Thanks.
We have one more question.
Yeah, so as you said, we learn from our errors, right? So when do you think you will have neuromorphic hardware that will work something like an FPGA and reprogram itself?
It's a fun question. I think that if it's fully programming itself, I don't know if we're anywhere close to that. I do think we want to have learning. I mean, in fact, these chips do have the ability to do learning. None of the algorithms I talked about really use that. We might have chips, or we might have algorithms in the not too distant future that hopefully are very brain inspired, that are intrinsically learning, and so the models put onto these chips will adapt themselves over time. So that would not be learning the same way as FPGA reconfigures itself. But in some ways, you mentioned FPGAs, these chips are very similar to FPGAs in terms of how we program them and how we think about them. So what you're suggesting is not impossible, it's just not really how we're thinking about it.
So the neuromorphic part, you have spiking, right?
Yes.
But just this Monday, I heard a talk by someone who used the connectome from the Flywire. And in that, she showed that there's a single spike that is induced that leads to a specific behavior in the fly, right? So that is very far from probabilistic,
Yes.
right? So I'm just curious. Yeah, you can solve, random walks. There's a lot of the central limit theorem is really helpful. But for such single spike events, right? That's hard to get.
Well, I'll say that these chips are rather conventional under the hood, right? So these are digital CMOS electronics. So if I want to turn off noise, I can turn off noise on the current hardware. I do think that the fly brain and the human and mammalian brains are quite different in terms of, I'm not aware in mammalian brains whether you have single neurons that have fully deterministic behavior the way that, that are perfectly well-defined connectomes. Like the fly connectome is very structured, right? Whereas our brains are much more randomly connected, right? So I do think there's some fundamental questions, but I'd say those are more computational neuroscience questions. We can probably implement either version of it in neuromorphic. But like that sort of neuron, I think it's the H1 neuron, I can't remember. It's been studied a while in flies, and it'll be interesting to look in our sort of, we haven't played with the model too much, to be honest, but to see how that works in that.
Thank you.
So just quickly, great talk. Very cool to see the Flywire, implemented so quickly. And the Brain Initiative has been collecting all sorts of other types of data, so including cell type data. We've released a whole mouse brain, cell type atlases, multiple thousands of different types of cells. So, the connectome is not just a homogeneous connection graph. It's connections between different types of cells that have different types of responses to neuromodulators, all that complexity that you're talking about. So, what do you see as far as how can you integrate that kind of data across different scales, cell type to connectivity, and how does that relate to the major obstacles you see for getting that arrow to keep going up and to the right in that complexity versus size plot, and how that relates?
So I love that question because if you looked at some of my original dentate modeling stuff, heterogeneity is fundamental to how the brain functions and is probably critical to understanding how disease interacts with the brain. It's not what neuromorphic computing is. We've done a lot of on neuromorphic, but a lot of the neuromorphic systems can handle that. It's just we haven't done it yet. Something like GPUs are terrible at heterogeneity. So I think that that's the place where neuromorphic might have an advantage, but there's some real work that's going to be needed to dive into some of this data and start to really, kick the tires on what we can do. So it's a fantastic direction to go.
Thank you so much, Brad, for telling us about how neuromorphic can help us understand neuroscience. And there is a WALS reception right now, so let's give a hand to our speaker.